The discovery of vaccines to prevent COVID-19 is great news. Never before have vaccines for a virus been discovered so quickly. But let's be realistic. Not everybody is going to get the vaccine, either because they refuse to or because it is not available to them. It is important that we continue to discover small molecule therapies to treat COVID and other viral diseases. Most of the approaches to treating COVID-19 have looked at modulating the immune response or disrupting the function of one of the virus's proteins, such as RNA polymerase. But little attention has been paid to the RNA itself as a drug target. SARS CoV-2, the virus that causes COVID-19, is, in its simplest form, an RNA delivery package. The virus binds to the ACE2 receptor and releases its RNA. The RNA represents the instruction booklet to make more viral copies using the host cell's own cellular machinery. Inhibiting or destroying the viral RNA would be the most direct way to kill the virus. The research group of Matthew Disney at Scripps Institute in Florida have developed a number of technologies to target RNA. RNA is a difficult target for a drug because it is a long, thin strand. They knew from other research that the SARS CoV-2 RNA requires a frame-shifting element to correctly translate the RNA into viral proteins. The frame shifting allows the ribosome to translate the RNA code into a protein, and then it has to shift back to make another protein. Several structural features of RNA enable frame shifting, including a piece of RNA that is folded back on itself, making a hairpin. This structure forms a defined pocket that can be exploited for drug binding. The research team designed an assay called Absorb Array, in which a small molecule library of 3,000 chemical compounds was arranged on an agarose-coated slide, with each dot representing a different small molecule. Then they added the RNA fragments, allowing them to bind to the small molecules. They washed away the non-binders, identified the dots where binding took place, and then deduced which fragment was binding to which small molecule. With this assay, they were able to identify a small molecule they called C5, which bound to the attenuated hairpin. C5 bound to the pocket in the loop, stabilizing its structure and inhibiting frame shifting. The ribosome could decode the RNA upstream of the frame shifting element, but then it could not read the RNA code downstream of the hairpin. The team went further and created what they called a RIBOTAC, a ribonuclease targeting chimera, not to simply stop the RNA's function, but to destroy the RNA itself. First, let's see what ribonuclease is and why it's important. Ribonuclease is a hydrolytic enzyme present in many tissues of the human body. It binds to RNA, including viral RNA, and cleaves the phosphate backbone, breaking up the RNA into small fragments. Now, back to the RIBOTAC. This is a hybrid molecule, or chimera, with C5, their RNA hairpin binder, at one end. At the other end, covalently bound through a linker, is a small molecule that attracts ribonuclease the way a lure attracts a fish. The C5 RIBOTAC seeks out its target, with C5 binding to viral RNA through the hairpin and ribonuclease binding at the other end. The viral RNA genome is destroyed, rendering the virus incapable of replicating itself. The C5 RIBOTAC was about 100 times more potent at inhibiting protein synthesis than C5 was by itself. What's more, C5 RIBOTAC is catalytic, or substoichiometric, meaning that one molecule inside an infected cell can destroy many strands of viral RNA, one after the other. The next step is to demonstrate C5 RIBOTAC safety and efficacy in a whole animal. Congratulations to the research team for this exciting and timely discovery to aid in our fight against viral diseases. For the sake of public health, please get the COVID-19 vaccine when it is available. Thanks for watching, and don't forget to subscribe to Science Sketch.